Okay, welcome to the last session. I'm Ma. I will chair this session. And the first speaker is, please go ahead.
Okay, thank you very much. So today's talk is actually maybe the last time I talk about morphogen gradients. Most of my lab does mechanics and tissue formation, but partly because G2 invited me and I'm working with G2 on morphogens, I thought I would talk about this. This is mostly actually stories which are finished. I've ordered the talk from kind of generally the newest to the oldest stuff. So if I run out of time, I'll miss out some stuff at the end, which it's been published for quite a while. So really, I've been interested in how morphogen gradients form, and in particular in recent times, also looking at how patterns and boundaries play a role in the patterning. So I'll show a morphogen gradient on the next slide. I know we have a more general audience, so I'll explain it clearly, but the first one that was discovered was bicoid. Bicoid is expressed in the anterior pole of the Drosophila embryo, and it forms a gradient of high concentration here and low concentration here. And bicoid is a transcription factor, and basically you have genes that respond to high bicoid and low bicoid at different positions in the embryo, and that gives you positioning. So in this case, wild type is on top, and on the bottom is a bicoid null. A bicoid null actually forms without an anterior. It forms with two posteriors, so it has an arse for a head. And normally I say you can choose your favorite politician to choose your name to describe it. I used to make a joke about a certain president, but I was told that was not allowed. Yeah, no, I thought you'd improve, Linda. And of course, this one dies, whereas the embryo at the top runs away. So this is what the bicoid morphogen gradient looks like in the early Drosophila embryo. It's highly expressed in the anterior. It diffuses through the embryo and forms this gradient, which actually fits an exponential profile remarkably well. So there's been many questions about how morphogens form, and in this talk, I'm going to try to run through quite a few of them. One is, of course, how does morphogen transported? If this talk generates any interest on your side, and maybe if you're coming from a very theoretical point of view, there's an excellent review in development from Alex Shear's lab a few years ago now, but basically going through the different models of morphogen gradient formation. And there's been a lot of argument about what could possibly be driving formation. And so I've been developing some tools primarily with bicoid to try to actually distinguish different models of morphogen gradient formation. An important point is that as a physicist, we often think, oh, biology, proteins diffuse, right? And it's nicely and simply done. The analogy here is given that ships, sailors who've been out at sea for a long time have got into port, they've just docked here, and all the sailors are coming off the ship, and because they've been at sea a long time, they're somewhat inebriated, and so they're staggering around, doing a random walk. But this isn't like biology. Of course, biology has cells and things that get in the way, and so you have hindered diffusion as sailors have to stagger around. And then, of course, biology is even more complicated, as in those proteins are binding to things and interacting, or in this analogy, you have pubs in the buildings, and so the sailors get caught at a pub and stay there for longer than stagger on. And so there's different ways of thinking about how particles move, and it's kind of important to distinguish that from sort of the classic physics experiments of, say, a dye in water. So what am I going to talk about? I'm going to mainly talk about how morphogen gradients form, and that's the work we've been doing recently. And then, depending on how much time, I'm going to touch on some of our older stories. Firstly, how is the readout of the gradient temporally controlled? So it's not just about putting things in the right position, it's also doing them in the right position at the right time. And then lastly, how embryos adapt to size changes. So first off, how are the gradients formed? This work was actually originally motivated because there's multiple models being proposed for how the bicoid gradient forms. The original one, the synthesis diffusion degradation, or SDD model, was simply that you have bicoid production in the anterior, it diffuses through the embryo, and then there's some degradation to control number. And that gives you a nice exponential profile, with the bicoid mRNA being localized at the anterior pole. Another model was that this is what's called a syncytium. It starts with one nucleus, that nucleus divides, it divides again. There's a single cell, which is the outside. but by the end of 13 rounds of division, you have 6,000 cells, and essentially the bicoid can be transported along the nuclei. Essentially, the nuclei shuttle the bicoid through the embryo as they divide. Another way is that rather than transporting the protein, you just have a gradient of mRNA. So you make your protein where you want it to be. And then finally, you could even imagine that the RNA itself diffuses through the embryo to set up a gradient, and then the protein again is made where you want it to be. And the key thing here is that all these models actually, if you use reasonably realistic parameters, generate a nice exponential gradient through the embryo. And so people use it to argue that all of these, there's been different papers arguing for these models. But if you look at, instead of asking what is the average gradient, but instead, what is the average age of a protein at a particular position from these different models, they have very different behavior. So the SDD model, protein is produced in the anterior, so that's where it's youngest. It diffuses out, so only old protein gets to the end because it has to it has to move across the embryo. It's similar for nuclear shuttling, except for you generally actually need to have, it's a slower process because you have to wait for each round of division. And so therefore, you tend to get a higher profile here. The RNA gradient, bicoid is produced everywhere. So pretty much the bicoid is the same age everywhere through the embryo, so it's pretty flat. And the RNA diffusion model is the inverse. So because the RNA is diffusing through, and you've got to wait for it to, the protein to then be made. In fact, it takes a long time for the RNA to diffuse to the posterior, so the posterior has the youngest protein. So although they have the same, if you just measure intensity, they look the same. The models all have very different predictions for how old the protein is as a function of position. And you can then ask, well, how on earth do you measure that? And so I was lucky to team up with Michael Knopp, he was actually interested in budding yeast. And his question was, when a yeast cell buds, does the new bud get the old protein or the new protein? Because the yeast cell doubles the proteins and then basically gives them out. And so he developed this timer where you essentially take a fast-folding fluorophore and a slow-folding fluorophore, and you measure the two channels. And of course, it's bound to your protein of interest. And essentially, if you see a green, then you've got young protein. If you see red and green, then you've got an old protein. And so we actually generated this. This is actually my only real bit of biology I've ever done in my life, was making this construct. We made a whole range of these, and you can actually then, after careful control, measure the ratio of the red to the green signals. And you can kind of see that the ratio is going up, and it, to be relatively quick, it turns out that actually only the SDD model can fit the observed ratio of the ages of the protein. It definitely shows that the RNA models are incorrect, right? Because the age of the protein doesn't fit. Sorry, the source diffusion degradation model. It's just the standard model, the things diffuse. So yeah, maybe I should just call this diffusion. Yeah, as an aside, and it's a relaxed conference, I think to a lot of physicists are like, duh, this is obvious. To a lot of biologists, they hate this diffusion model. I've had a very eminent biologist tell me that biology is ordered, and that he believed in diffusion across the synapse of the brain, like because it's like 10 nanometers. But beyond that, you think, no, that can't be possible. And this is why people believe in things like cytonemes and all sorts of other approaches. Cytonemes are real, by the way, but just I think there's a natural inclination against diffusion, because when they hear that it's random, people suddenly think, well, if it's random, how can it work? I think most physicists can work that's not a problem. But what's nice here is that it shows that the diffusion model certainly fits the best. And we could do some nice things, like we could follow the total fluorescence, so essentially how much protein there is versus the ratio of the signal, and across multiple stages, multiple embryos, so from very early to young, and we could actually nicely map how the system behaved, and in fact see when and where production was turning off as well, because later in the nuclear cycles, bicoid's only needed at the start. So after it's done its job, it's got rid of, and so actually that's why you start to see this decrease in intensity, but the ratio keeps going up because you've turned off production, you've only got old protein left. And the amount of old protein is decreasing, but the ratio increases. So this was published now quite a few years ago. But in more recent times, we kind of wanted to follow up on, well, that gave pretty strong evidence that diffusion was driving the system. But can we look at whether, is it as simple as that? Is it the diffusion is uniform across the system? And the idea really here is that, going back to this analogy, you've got sailors going around, they're drunk, they get to the bars, but of course, the bars nearest the boat are going to get full quickly. And they don't want to stay there because it'll be too long to get a drink. So they'll stagger on a bit further and stagger on until they can find a bar which is empty and they can go and get a drink. So the idea here is that you might well have concentration-dependent dynamics in your system because essentially of the protein crowding. And so essentially we wanted to ask, is there spatial inhomogeneity in the Bicoid transport? So to do that, and this is work actually started by an undergrad student who's now doing a PhD at Harvard, was basically do FCS, so fluorescence correlation spectroscopy, just in the anterior of the embryo, where everyone always used to do it anyway, because that's where there's a nice signal, but also in the posterior. For those that know about FCS as a technique, it actually works better at low concentrations. You want to be just above the background noise. So in fact, the FCS curves are better here than they are here. But I think intuitively, people don't like to do that. And so we measured that, and somewhat surprisingly, when we looked at Bicoid GFP in the anterior or the posterior, the effective diffusivity was about twice as fast. So yeah, Bicoid, which is at 400 microns from the anterior pole, is diffusing far quicker. If we do it with just a simple nuclear localization signal with an EGFP, there's no difference. And so this was very exciting. I actually didn't believe this result. This was, say, it was collected by an undergrad, and I was like, no, this can't be true. But it's been repeated now by 2 different people, and actually 2 other labs have also seen it. And so I'm pretty confident that it is a real result. So then we wanted to ask, well, what on earth is driving this? And so we focused on the Bicoid homeodomain, which is actually known to have binding sites to both DNA, but also to cytoplasmic elements. And what we then was we created a range of different mutants, and it's a bit small here, but one of them, for example, in the nucleus, if we interfered with the binding of Bicoid to DNA, which is this mutant here, the N51, we see an increase in the diffusivity in the nucleus, which makes sense. If it's a transcription factor which can't bind as well, it's going to stay more free, it's going to move around. So this was kind of nice that we could actually do direct mutate specific sites in the protein and look at how it changed its dynamics. And this kind of gave us the clue that the homeodomain is important. So we kind of took the alternative view, which is rather than trying to dissect piece by piece the homeodomain, let's take the whole homeodomain and stick it on NLS. So this is just a nuclear localization signal, and we now stick the homeodomain on it. And it turns out that if you do this, that now the NLS actually behaves like the Bicoid does, so the diffusivity gets a lot smaller. And in fact, the gradient, the black is the wild-type Bicoid GFP, the green is just an NLS, but with the Bicoid homeodomain attached to it. And then the pink is just without the Bicoid, without the homeodomain. And so what we can see here is that we can actually now start to dissect this protein and say which parts of the protein are actually regulating the dynamics. Sorry, that clicking is kind of annoying. What we're trying to do, if we get a grant that I've just put in, we're actually going to try to more carefully dissect this and try to get at the specific elements that are driving this. So you can actually, if you want to be a bit cruel to yourself, write down some models for this. So what we're basically arguing is that we have positional dependence in the diffusivity. So if you go to your standard reaction diffusion model, you now can't just have the diffusion outside, you have to have it inside the first part of the derivative. And then essentially you have this is the slow form and the fast form. And so you have the different diffusive terms, the protein can degrade, and then you can basically switch between the slow and the fast forms. We can actually measure all of these parameters except for the beta, which we kind of end up being a single fitting. But what's nice is that you can throw that in and actually fit the data very well, including positional diffusivity actually gives you a better fit to the data, particularly towards the posterior end. So normally when you just have a simple exponential, it actually starts failing towards the posterior, but if you include the fact that the protein can diffuse faster, it actually does a lot better job. And so this was published last year, and I was very pleased that it was actually voted as one of their 3 best papers of the year, which was kind of nice for Tamarai. What am I doing on time? 9 minutes. Okay, good. So that's not too bad. So then, to me, this is pretty conclusive. But as I said earlier, there is kind of a tendency that people don't like diffusion. And so people have proposed different models that have basically the production of bicoid to be spread through the system. So this is the paper that sort of really first proposed this, where it proposed an RNA gradient. Thomas Gregor actually published a few years later a paper where he analyzed the distribution and claimed opposite results, but essentially they're using the same technique, which is fluorescence in situ against bicoid RNA, and you can have all sorts of arguments about who did background correctly, who did the controls correctly. So we wanted to take basically a different approach. Can we use a different tool to try and ask the question, where is the protein actually made? And so the tool that we turned to was SunTag, which has become extremely popular recently. So it was first developed over 10 years ago, but it's really taken off the last few years. So this is a tool for actually observing protein translation through the ribosome. Essentially, you tag your protein of interest with this SunTag, which is just a piece of DNA. And then as it goes through the ribosome, essentially as it comes out, the SunTag is detectable by this antibody, this GFP, which can be genetically encoded. And so essentially, as your RNA moves through the ribosome, you basically start seeing these green spots come up as it binds. And you can check that it's actually real because you can also then look at the mRNA, and when you have the mRNA and the SunTag overlap, you know you have translation, and where you just have a dot, that's just a piece of RNA. It's not in a ribosome being translated. And so Munier-Lager in Montpellier invested a lot of effort to put this into in vivo. And so although it had been used in cell culture for a while, there's a number of challenges why it's not trivial to get it working in vivo, but she managed to succeed. I had a very nice paper a few years back where they could actually observe translation live inside the Drosophila embryo. So we went to Munier and said, well, can we do this with bicoid, please? You know, because there's this big question about where and when bicoid is actually made. So we made a bicoid SunTag line, and this is the in situ. Bicoid is in the anterior side. This is not super clear. We should probably outline where the embryo is. And this is the antibody against the SunTag, and you can see nice right in the anterior pole. And so this is, and we can then do the comparison. Again, it's not super great on this slide, but you essentially can see clear localization of the spots. And sometimes you don't have localization with the bicoid, but that makes sense because not all mRNA is being translated at the same time. And then we could actually then look at the number of how much bicoid is being translated, and unsurprisingly, in the anterior it is high, and in the posterior, actually, we basically didn't see anything above noise. We saw some correlations, but it was nothing beyond what we would expect from just random fluctuations. And so we could actually then look at the translation across time. So this is really just focusing on the posterior pole, but you can see nice high translation early on during embryogenesis. Essentially, we worked with Tim Vile at Cambridge, who's an oogenesis person, so looks at how the egg is made. And what he showed as part of this project is that there's no translation in the oocyte, and it's quite interesting how that's stopped. But as soon as the egg is laid, that inhibition is released, and you start seeing translation. But then that translation starts to turn off. So once bicoid has done its job, the production comes down. So we can start to now actually really get a handle on the production rate of the protein in vivo. And so there you can see the percentage production rate going down over time. Well, that's the mRNA, but it's the same for the translation. And so this is a very nice tool. So it's an independent way. It doesn't rely on in situ hybridization to actually verify that translation is only occurring in the anterior. And also, this was very much unknown about when and where bicoid was being turned off. And there were quite a lot of different arguments, but people only had access to the intensity of bicoid, which actually isn't helpful for telling if production is there or not. And so again, you can throw this into a model. We can basically take our data and we know what the bicoid RNA distribution is like. We know the translation turns on after egg laying. We actually also know, partly from this project, from Tim Weil's work, that the rate of degradation of the RNA increases in nuclear cycle 14, so we can put that in. And then we model the diffusion as being some sort of linearly related term. In fact, yes, sorry, that's not wrong. This is not going to 0 here. It's like starts at 4.5 and then goes up. And you can throw this all in, and actually you get a very nice exponential fit to the gradient, which is what is observed, which is kind of your default. But you can really start to describe the gradient formation over time in more detail. And so this is currently on bioRxiv, and hopefully going to get nice reviews. So that's all the new stuff. I have 4 minutes, so I will talk about some of the older stuff just to kind of complete the story. So the first part, really, how a gradient's formed, at least for the bicoid gradient, it really is diffusion. It's quite hindered diffusion, and the diffusion varies in different parts of the embryo, which I think is really interesting, and we don't fully understand the mechanism behind that. And then translation really is anterior localized. It's not through some mechanism whereby somehow the protein is being made here. And so basically the original source diffusion degradation model is qualitatively correct. It is quantitatively incorrect, but it was a good estimation to begin. Yeah, so the next question is, of course, well, it's one thing to have a morphogen gradient, which has some concentration-dependent readout, but how does it actually specify at particular times, at particular positions? And actually what we were really interested in with my first student, Anshi, was how is it temporally coordinated? As you saw on the previous slide, the production goes up and down, the gradient is actually changing continuously. So how is this controlled? And so I'm going to skip this. So what Anshi did was she actually built an optogenetic bicoid where by shining light, we could turn off the ability of bicoid to be act as a transcription factor. Essentially what the light shining did was it forced it to cluster on the DNA. And by doing that, it inhibited its action. So we put in our optogenetic bicoid. This is normal, so our construct in the dark could fully rescue the patterning. But if we shine light, then basically this Cry2 domain folds over, blocks the bicoid from being read out by the polymerase, and then you essentially end up with a mutant phenotype. So for example, you lose the hunchback anterior stripe. So the nice thing about this is that we could then do, so I'm just going to skip this because I don't want to overrun. I hate it when people overrun. So what we could show is if we shine light, for example, we can stop translation, the bicoid action. And so, yeah, this is what I wanted to get to. So what we could do is, what Anshi did was a nice series of experiments where she put embryos in and she exposed them to different levels of light relative to gastrulation. And so these are nice structures which we can observe in the embryo about 2 hours after gastrulation. And what she could do is if we just inhibited for 10 minutes before gastrulation, we lost the most anterior pieces, so we lose the mouth. If you do half an hour, then we actually lose the neck as well. And as we increase the level of time, we decrease the number of stripes that get specified. So the really interesting thing here is that the inhibition of bicoid, the impact is from the anterior where the concentration is highest. That's actually where the biggest impact is, not where it's normally lowest. And so we could actually, yeah, so basically the key point is that the anterior genes are actually more sensitive to loss. And this has kind of been shown in other systems now that, for example, with DPP and the wing disc, that morphogens are most sensitive to where they're actually at their highest concentration, because essentially those genes are, have the weakest binding sites, really. And so, in fact, that's why they react most strongly to when you perturb the system. So we could actually build up this sort of temporal patterning where we could see over what time period did we require bicoid to be active in order to actually give the correct structures. So for example, some of the most posterior ones actually only needed it for a very short period of time in order to be successfully positioned, whereas the most anterior ones needed it for an extended period of time, for almost an hour and a half, in order to be correct. So basically, just to conclude, the synthesis diffusion degradation model is very, seems like the most consistent way of describing gradient formation. We used two different approaches. We measured protein age, and we used fluorescence correlation spectroscopy and SUNTAG, and all basically support this as a good qualitative model, so long as you make some sensible adaptations. And then in the second part, just showed you that really the timing of readout is important. Biological systems don't respond in a uniform way across their field of range, which is how 20 years ago, a lot of the work kind of assumed that. And so got to be careful. And in the last part, I basically talked about scaling, but I didn't have time to cover. So Tamarai did an awful lot of the work in the first half of the talk. And then Anchi, with help from Chris, did the work on the optogenetics. And thank you for your attention.
Thank you very much. So let's move on to the questions and comment.
First part where you showed spatially varying diffusion. Can you go to the equation? I didn't understand what you meant by the switching of the two things.
What we see is that
Sorry, before you go, the first equation, second line, that should be a rho f, right? Otherwise, it doesn't make sense.
There is here.
No, the conversion. Otherwise, there's two terms.
This is the slow going to the fast.
Okay.
And this is the slow. This is the fast going to the slow.
Okay. Okay. So what's the conversion? What is physical?
Well, so this is I should have put the four equations actually make sense. What we see is, do I have it here in the data? When we do the FCS, we see two populations in the cytoplasm, and we see two populations in the nucleus. We see a fast form and a slow form, and we actually have the percentages of that from the FCS. So what I'm actually, so I didn't explain this very clearly. This is a model in the cytoplasm because that's forming the gradient across the whole embryo. And essentially we have the slow portion and the fast portion, and we have a switching between the two. You can get away with just a single effective one, but which, I should have probably done here. I should have either shown one or four. This is not helpful. Yeah.
So in terms of the space dependence of the diffusion coefficient, if you explain where that comes from, I didn't quite understand. But at some point, it sounded as though you're trying to explain it in terms of crowding effects, in which case it shouldn't be space dependent so much as concentration dependence, right?
So in the paper, we only measure FCS in two positions, posterior and anterior. There is, as far as we're aware, no difference in the cytoplasm between the anterior and the posterior. So yes, I am 95% certain this is not an X, but this is a rho. This is a concentration. And again, I put in a grant where I've asked if we can have someone to actually do the FCS every 20 microns, build it up, then correlate it with the concentration. But yes, so for this sake, I'm certain it's a concentration-dependent effect. But for the paper, we only had measurements at like two locations. So where's the data? Yeah, we had anterior and posterior. And so I felt it was a bit too pushing to, so we say that it's probably concentration, but yeah, that's where it comes from.
So I guess this is at the stage where it's before the cellularization.
Yes.
Right? And so you didn't mention anything about the degradation rate.
Yeah.
So one is, what is the origin of degradation, and could that be space dependent?
So the only bit I touched upon degradation very briefly here, which is, this is quite a hard graph to read, but essentially this is how much protein I have. And this is how old the protein is. So younger here, older here. So early on, you don't have much protein and it's young. You get accumulation of more and more protein. And as you accumulate more and more protein, the average protein age gets older. And then actually cellularization is here. So this is nuclear cycle 10, and then this is cellularization. And then after cellularization, essentially you stop production. And we actually see that in the SUNTAG, that the production stopped. So this is degradation. So essentially you have no new protein. So the existing protein is getting older, which is why we're continuing upwards. But as degradation happens, of course, our amount decreases. But we can estimate it to be around, what did we estimate it to be? I think it was around 25 minutes, the lifetime. It's in the order of 20, 30 minutes. It is also not clear whether degradation rates change. And it may be, there is some evidence for that, but the tandem is good for looking at global behavior, but it's not very good for getting that level of specificity. So yeah, we have a really cool tool for dynamics. We have a really cool tool for production. We don't yet have a really cool tool for degradation.
And the two species of bicoid was because it's bound or unbound.
Yeah, and basically you have bound and unbound in the cytoplasm, which we think is binding to cytoplasmic elements like actin. And there is a putative actin binding domain on bicoid. And in the nucleus, it's easy to understand. It's a transcription factor. It's either bound to DNA or it's not. So that's the fast and the slow.
It makes me think of macromolecular crowding and some way of setting up a gradient across the thing that is more concentrated in the anterior and less in the posterior. And is there a way, like, are there compatible solutes that are less of a barrier to diffusion that would be higher concentration in the posterior that could, in part,
that's
you know,
I can't answer
your result.
I can't answer specifically, but there was a classic experiment by Thomas when he was a PhD student where they injected beads into different sized embryos. So they took one species lays embryos which are like 1.5 millimeters long, like they're a big fly. And they actually see that in the bigger embryos, the diffusion is faster of these beads. So they're more inert, they're not interacting. And I think their argument there is there was more space, and so there was less crowding, right? So I don't think in that paper they actually did different concentrations in the same sized embryo, which maybe you might start to actually see crowding effects. We're really not sure if it's crowding effect or whether it's actually a binding effect to specific elements in the DNA and in the cytoplasm, which it could be either or both.
Right, and so you're saying it may be making interactions with actin. Is actin more abundant in the anterior than the posterior?
No. And that's why, to come back to the previous question, I didn't want to say that it was I had it as a position dependence, but like in reality, I think it must be something to do with the local concentration. Yeah.
And is it traveling as a monomer?
That's also a very good question. Now, a principle in FCS, you should be able to see dimerization and so on.
But is all of your protein labeled?
Yes. So the beautiful thing about Drosophila is this has only got the bicoid GFP. There's no native bicoid. So again, aim 3 of my grant, which I'm not going to get because success rates are so lousy, but there is actually some nice new approaches, scanning FCS, where you can look basically at multiple points rather than at a single one. By looking at the correlation between the multiple points, you can actually see kind of a better estimate of how big the structures are, and then you can get an idea of the oligomerization. So yes, I hope to answer that question, but depends on UKRI. Okay.
Good.
Thank you.